Hi and welcome everyone to this Biopen webinar, and thank you all for joining. We are very honored here today to have Veronica Jové from the Rockefeller University with us here today. And Veronica will be presenting her work on the neurobiology behind the taste of blood in mosquitoes.
Okay, good morning everybody, or I guess good afternoon depending on where you are. I hope everybody's hanging in there okay given the current circumstances. We are surviving here in New York City. So today I'm going to share with you my work on Aedes aegypti mosquitoes. And so from our human perspective, a mosquito bite is a global health threat transmitting diseases like Zika and dengue. But today I'd like to share with you the perspective from a female Aedes aegypti mosquito. Once she lands on an attractive human like you, how does she recognize the taste of your blood? And so when a female bites, she uses the dedicated blood feeding appendage known as the stylet to pierce skin and pump blood. The needle-like tip of the stylet is in direct contact with blood as the female feeds, and here we can see the stylet in action from the perspective of underneath the skin. Today, I will show you how this unique sensory appendage is specialized to recognize the taste of your blood. So first off, only female mosquitoes feed on blood, and female mosquitoes will only feed on blood when they need the protein from your blood to develop eggs. The rest of the time, female, like male mosquitoes, feed on nectar as an energy source. And although both blood and nectar are appetitive stimuli for the female, her feeding behavior when she feeds on blood is actually quite distinct from when she feeds on nectar. And so one key difference between the two is the feeding appendage that's used. So while the stylet is in direct contact with blood during blood feeding, the nectar feeding appendage, the labium, remains on the surface of the skin and never directly contacts blood. In contrast, during nectar feeding, only the labium is in direct contact with the nectar meal. Another key difference between the two is meal size. So female mosquitoes will reliably engorge on blood, which means that they'll double their body weight on the blood meal in order to obtain enough protein to lay a full clutch of eggs, whereas the sugar meal is much smaller in size. And finally, the blood meal bypasses the crop, which stores the sugar meal, and is immediately directed to the midgut, which is a digestive tract specialized to digest blood proteins. And so we think that this strict segregation in feeding programs is really important because it may allow the female mosquito to maintain her metabolism by taking these small carbohydrate-rich nectar meals without diminishing her appetite for the large protein-rich blood meal necessary for egg development. And this ability to maintain two mutually exclusive feeding programs for carbohydrate-rich and protein-rich meals is an unusual feature, and we were really interested in understanding what is the relevant information that allows the female to select the appropriate feeding program. And so we started by comparing three key differences between the two feeding programs, and starting with the difference in nutritional value. So to ask if blood proteins are required to initiate blood feeding, we can use an artificial feeder like the one pictured here. And so in this assay, a thin piece of parafilm separates the female from the meal, forcing her to pierce through the parafilm with her stylet in order to contact the meal in the same way that she must pierce through skin with her stylet in order to contact blood. And so if we present attractive human cues like heat and carbon dioxide in this assay, female mosquitoes will reliably engorge on blood. However, if we maintain the same attractive human cues and instead substitute in the protein fraction of blood, which is sufficient and necessary for egg development, females will not engorge. And surprisingly, researchers in the 60s found that a protein-free solution of key plasma components like ATP, sodium chloride, and sodium bicarbonate are sufficient to promote engorgement, letting us know that the protein requirement for egg development is decoupled from the initiation of blood feeding behavior. So this led us to believe that chemosensation is important for initiating blood feeding. And so we first asked if the presence of attractive human cues like odor, heat, and carbon dioxide is sufficient to promote the mechanics of blood feeding behavior on any appetitive meal or if there's something really special about the taste of blood itself. And so in order to ask this question, we maintained all of the attractive human cues in our assay, but substituted out the key components of blood for the key components of nectar. And so one possible outcome of this experiment could be that female mosquitoes will engorge on the nectar meal and send the meal to the midgut as if she were feeding on blood, because the presence of human cues is sufficient. Another possible outcome was that the sweet taste of the nectar meal could be hardwired to the nectar feeding program. And if this is true, we would expect the female to take a smaller meal and direct the meal to the crop, even though the human cues are present. But actually, when we performed this experiment, to our surprise, neither of these outcomes occurred. And instead, what we found is that female mosquitoes did not drink any of these sugar meals. And so this tells us that human cues are not sufficient to promote blood feeding behavior, and that there is something special about the taste of blood itself. Furthermore, nectar feeding, or the taste of nectar, is not hardwired to nectar feeding behavior. And once the female pierces with her stylet, she's selectively searching for the blood meal, will not accept nectar as a substitute. But at the same time, if we present all of these same sugars at the same concentrations in the nectar feeding assay, now all of them are sufficient to promote feeding. So therefore, this response to nectar sugars must be flexible, and it also must be context dependent. And so one key difference we identified between these two contexts is a difference in feeding appendage. So in the blood feeding assay, similar to when a female is biting a human, the stylet is the only sensory appendage in direct contact with blood underneath the skin. And in the nectar feeding assay, similar to when a female feeds on nectar, the labium is the only sensory appendage. Well, the labium, but not the stylet, is in direct contact with the nectar meal. The legs are also in contact with the nectar meal. But we focused on the comparison between these two mouth parts because the legs are not used to ingest the meal. Okay, so together all of these data led us to hypothesize that the stylet is not only able to recognize blood, but it's somehow able to distinguish blood from nectar to assign this context-specific response. And of course, the important question here is truly how could this be happening? And so in order to identify stylet neurons that could be involved in detecting blood, we compared the female stylet to the male stylet, which exists but is never used in normal male biology since male mosquitoes do not feed on blood. And we identified a number of female-specific neurons, and importantly, we found that these sensory neurons send their chemosensory dendritic projections all the way out to pores at the very tip of the stylet, perfectly positioning them to come into direct contact with blood. And so we next wanted to know, well, what do these stylet neurons respond to? And so in order to answer this question, we developed with our collaborators a transgenic mosquito that expresses the reporter of neuronal activity, GCAMP, in all neurons. And shown here is our ex vivo stylet imaging preparation, in which we can simultaneously capture activity from all stylet neurons in just one imaging plane, since the tissue itself is so thin and small. So as a proof of principle, if we depolarize all neurons by adding a high concentration of potassium chloride, we see we can reliably detect robust responses across the entire population. We can quantify these responses and plot them as a heat map moving along the length of the stylet from proximal to distal. And so in these heat maps, each column represents the responses of one neuron, and each row represents the responses across the neuronal population to one ligand. And so in the first column, we have the responses to water, which is our negative control, and you see that we detect no changes in fluorescence. Whereas in the second row, we have the responses to our positive control, potassium chloride, which you can see we detect responses across the entire population. Okay, so now this was super exciting that we finally had a way to really capture responses of stylet neuron activity, but the next challenge that was presented to us was that blood is an opaque ligand. And so returning to the anatomy, what we needed was a way to restrict blood delivery to the tip of the stylet where the chemosensory pores are, without having it interfere with our ability to record changes in fluorescence in the cell bodies along the length of the stylet. And so this is where the Biopen comes in, and I'm super grateful to Gavin, who flew all the way from Sweden to New York City back in the days, the good old days when we were allowed to travel, to come set up a custom way to deliver blood to the stylet using the Biopen. And so actually at this resolution, you can see individual blood cells. And I think I might have forgot to mention that the area we were trying to restrict the blood to before it got to the cell bodies was really just a buffer of about 20 microns, something that's really difficult to do with any perfusion technique or any of the millions of other things that I tried before. And so we're super excited to finally be able to do this experiment, which is to deliver blood to the tip of the stylet and measure the response to the entire population, or measure responses in the entire population to blood. And what we found is that approximately 50% of stylet neurons respond to blood, and that these neurons are able to consistently respond to multiple presentations of blood. And so here we've plotted these responses as a heat map, and in this heat map contains the responses for one individual female. Now, if we compare across multiple females, we see that stylet neurons do consistently respond to multiple presentations of blood, but that the exact number and position of blood-sensitive neurons is not stereotyped across individuals. And so this led us to ask, well, how exactly is the taste of blood being encoded? We weren't even sure if all individual females are picking up on the same components of blood. And this is really complicated by the fact that blood is this complex and variable potpourri of ligands that span multiple canonical and non-canonical taste qualities. So some components of blood that we're familiar with, like sodium chloride and glucose, are typically associated with canonical taste qualities like salty and sweet, but others like ATP, which definitely has a powerful behavioral effect on the mosquito, is not really typically associated with the taste quality, and we don't even have an understanding of how this is detected molecularly in invertebrates. And so it was an open question whether or not these taste qualities were represented in blood and if there were multiple. And if there were multiple taste qualities that form the perception of blood, we wondered, would this follow a labeled line model that has been derived from years of work in Drosophila, rodents, and humans, where distinct taste qualities are thought to activate distinct populations of taste neurons. And so in order to answer these questions, we needed to simplify blood into a chemically defined mixture that we could ensure was reliably the same across all individuals. And so to do this, we selected 4 components of blood that have been shown behaviorally to affect the probability of initiating blood feeding behavior. When we delivered this mixture of 4 blood components, which we call Mix+ATP, to females in our artificial feeder, we found that both blood and Mix+ATP reliably lead to engorgement. And so we next delivered both meals to the stylet in order to measure neuronal responses. And what we found is that both of these meals activate the same subset of stylet neurons. This was super exciting because now we have a reliable way to activate the exact population of neurons that we're interested in. And so in a new set of animals, we went back and we presented each of the components of Mix+ATP individually. And then we performed hierarchical clustering based on the functional responses to these individual ligands. And we found that blood-sensitive neurons clustered into 4 distinct groups. And the first 3 groups could be activated by a individual component, so ATP, sodium bicarbonate, or sodium chloride, respectively. But what was interesting is that 4.5 millimolar glucose was not sufficient to activate any stylet neuron. And it was also weird that this 4th cluster of neurons was not activated by any individual component. We then went back and we started dropping things out of Mix+ATP, and we found that this fourth class of neurons is activated by a mixture of glucose, sodium bicarbonate, and sodium chloride. And so we term these neurons integrator neurons because they're responding to this mixture of blood components. So keep these neurons in mind because we'll return to them at the end of the talk. Okay, so now we have an idea about the fact that the taste of blood spans multiple canonical and non-canonical taste qualities, but let's kind of return to the original question that I posed at the beginning, which is how is the stylet distinguishing blood from nectar? And what is the key difference in sweet taste processing that's underlying this clear difference in behavioral output to the same ligand depending on the context? And what we were particularly interested in is the responses to glucose, because glucose is the only sugar found in blood, and it's also a principal component of nectar. And so to address this question, we first performed an RNA sequencing experiment on the female stylet, the male stylet, and the female labium. And what we found is that the female labium, but not the female or male stylet, expresses all of the predicted orthologs to the sweet taste receptor family. And we decided to focus in on one of these in particular, GR4, because it's the ortholog to the two most ubiquitously expressed sweet taste receptors in Drosophila melanogaster. We then created a driver line for GR4 and confirmed that it is indeed expressed in the labium and not the female or male stylet. Another motivation for creating this GR4 driver line was to be able to ask if activation of these neurons is sufficient to drive nectar feeding. And in order to fully answer this question, we also needed to develop a chemogenetic system in mosquitoes. And this system takes advantage of the fact that Aedes aegypti mosquitoes do not have a baseline behavioral response to capsaicin, which is the endogenous ligand for the vertebrate ion channel TRPV1. And so theoretically, if we selectively express TRPV1 in GR4 neurons, then by delivering capsaicin, we should be able to selectively activate these neurons. And so here I'm showing you the genotype controls to show that all three genotypes have a normal baseline behavioral response to sucrose and water. However, only in the females that express TRPV1 in GR4 neurons do we see an increase in feeding when we add capsaicin to water. And so this led us to conclude that female mosquitoes do have a canonical sweet taste pathway and that it's being utilized in the labium, but it's notably missing from the stylet. And kind of a related conclusion is that although activation of this sweet taste pathway is sufficient to drive nectar feeding, when sweet taste pathway in the labium is sufficient to drive nectar feeding, it's not expressed in the stylet. So the stylet, neuronal activity in the stylet, therefore, is dispensable for nectar feeding. Okay, so if the stylet is specialized to detect blood over nectar-specific sugars, we would expect that the stylet would not respond to behaviorally relevant concentrations of sucrose and fructose. And so to answer this question, we return to our original GCAMP imaging preparation that I introduced to you earlier. And when we delivered these concentrations of sucrose and fructose, we did not find any stylet neurons that were activated, although all of them responded robustly to our positive control of high concentrations of potassium chloride. And so from these data, we concluded that the stylet is indeed specialized to detect blood over nectar-specific sugars. But what about glucose? And as I mentioned to you, this is the most interesting sugar because it's the one commonality in between blood and nectar. And so to address this question, we returned to our clustering analysis that we performed earlier. And as I mentioned, 4.5 millimolar glucose, which is roughly the concentration found in blood, is not sufficient to activate any of the stylet neurons when presented alone. But we were interested in these integrator neurons because they respond to a mixture that includes glucose in addition to sodium bicarbonate and sodium chloride. So we decided to further dissect this response, and what we found is that although 4.5 millimolar glucose alone is not sufficient to activate integrator neurons, it is being detected because when this concentration of glucose is co-presented with sodium chloride or sodium bicarb, now it is sufficient to activate integrator neurons. We found that the greatest response was detected in the presence of all three. And so, therefore, this single class of sensory neurons is able to integrate across three distinct taste qualities to assign blood context-specific information to glucose. And polymodal integration across taste qualities was really an unexpected finding because in Drosophila, rodents, and humans, it's thought that distinct taste qualities activate distinct subpopulations of sensory neurons. And so we propose that in the mosquito, this unique taste coding system is just another level of specialization that allows the female to selectively recognize blood over nectar. And to summarize, what I've shown you today is that these four components of blood are sufficient to recapitulate the behavioral and neuronal responses to blood, and that the stylet is specialized to detect blood over nectar. And we think that this is a really remarkable example of how specialization in sensory neuron coding underlies the discrimination between these two essential nutrients that each promote a mutually exclusive feeding program. And with that, I'd like to thank my PhD advisor, Leslie Vosshall, everybody in the Vosshall lab, and specifically Jiayan Gong and Trevor Sorrells, and also our collaborators who helped generate the pan-neuronal GCamp mosquito that I introduced to you earlier, and also Gavin from Fluicell, who helped set up the Biopen, and our other collaborators. So thank you.
I just want to start with some questions for you, just out of curiosity. How did you manage to do the experiments with the mosquito stylet? Because I can imagine that it's quite fiddly.
Oh gosh, yeah, this is the part that gets glossed over. It's a very tiny structure. It's incredibly difficult to work with in terms of being able to manipulate it and make sure it's flat so that you can get all of the neurons in one plane. But that is what 6 years of a PhD are for, I guess, for perfecting that technique. So, yeah.
Okay, so another question. Now you've sort of you're done with this step or this part of the work. What's next? How do you build upon this? What's the continuation?
Yeah, this is a great question. I guess I don't know how interested people are, but I have a couple extra slides. Should I include them or?
Yeah, sure. I think we have a little bit of time, so you can go ahead and take over the screen again.
Okay, I guess you have to stop.
Yeah, I'm gonna stop sharing.
So one question that maybe some of you were thinking is activation of the stylet neurons sufficient to drive blood feeding behavior in the same way that we were able to use the TRPV1 experiment with the labium-specific driver and drive nectar feeding. And so I think this is a really exciting next step. We've made some progress towards this, although we haven't fully solved it. But in that RNA sequencing experiment that I showed you, in that context, we were looking for the transcripts enriched in the female labium, but of course with this data set you can do the opposite as well and look for things enriched in the female stylet, and not only these two tissues here, but we were able to compare it to a much larger transcriptome data set generated by a postdoc in the lab, and so we identified transcripts that are truly expressed only in the female stylet and not anywhere else in the mosquito. Because we have no idea which genes are involved in detecting blood, and so something that was specific to the female stylet was our best guess for what might be important. And so when we created these driver lines, we were able to confirm that we did selectively label sparse subsets of female stylet neurons only in the female stylet and not in these other tissues as predicted from the RNA-seq. And when we looked at the mosquito brains, I didn't get a chance, sorry, to introduce the brains, but this lower part of the brain here is where all stylet neurons project to, and so we're seeing that we've labeled a sparse subset as predicted, but what's really cool is that there's no expression anywhere else. So we really have succeeded in making these specific driver lines, and what we found was that these neurons label the sodium bicarbonate sensitive population for one of the driver lines, and for the other one, only half of the neurons are blood sensitive, but that half labels the integrator neurons. And so we're still working out, there's a lot of very silly technical reasons why we just haven't been able to do, including COVID, but why we haven't been able to really parse this out. Although I have to tell you, my guess is that I don't think these are the perfect driver lines to answer the question. Ideally, we'd have something that labels a larger population of blood-sensitive neurons. But this is kind of, I think, where the future directions are going.
Okay, great. One other thing that sort of I wonder a little bit about is so what. How do you use this knowledge? What can, I mean, what implications does this have for, let's say, if you want to prevent mosquito-borne diseases or things like that? That's understanding their feeding behavior.
Yeah, great question. So our lab currently is focused mostly on the basic biology, the neurobiology underlying sensory detection of mosquitoes, but there are a couple different things that you could imagine. So the first is that mosquitoes need to take a blood meal from an infected person in order to become infected themselves and transmit diseases. So if you can somehow prevent mosquitoes from taking this initial blood meal by knowing what are the requirements for initiating blood feeding, you can imagine that you could basically drive a wedge in the entire cycle. But the other thing that can be exploited, that already has been exploited, is by knowing what the females find appetitive. Now you can go in and add drugs and different compounds that might affect their behavior later, right? But you're only getting them to drink it because of the fact that you've added these appetitive ligands. So this mix of ATP and sodium bicarbonate and sodium chloride, there's a postdoc in the lab, we've published a paper, I believe last year, showing that if you add specific compounds to this mixture, mosquitoes will engorge, they love it, and then their biting is suppressed afterwards.
Yeah, super. Very, very, very exciting, I think.